We really needed like 10 to the 6-fold host depletion, so 99.9999% removal of human DNA to get that ratio down from 1 billion to 1 down to somewhere around 1,000 to 1, which might work. So I took on a PhD student early in my time at the University of East Anglia in my first year in 2013. And his name is Solomon Magwisya. And Solomon started working on methods for depleting human DNA in blood samples. And that is how we came across ArcticZymes. We were at a conference in Munich, in Germany, the qPCR conference. And Solomon went up to the ArcticZymes booth and he asked, do you have any enzymes that might work in whole blood to deplete or to digest human DNA? And the person on the booth said, you should try HL-SAN. It's our salt-activated nuclease that is heat labile. We tried it out, and after a while we got some good results, but there's a little bit of a story behind that. Solomon and I were looking at different ways to deplete host nucleic acid in samples. And we'd come across a few different ways. There was a mol lysis kit out from Malzahn. There was a New England Biolabs kit out that removed host at the DNA stage. But none of these methods were giving us sufficient host DNA depletion. We really needed like 10 to the 6-fold host depletion, so 99.9999% removal of human DNA to get that ratio down from 1 billion to 1 down to somewhere around 1,000 to 1, which might work. So we weren't achieving that with the commercial methods that we found, so we went around developing our own. And what we did is we tried different things. We used differential centrifugation. We used different enzymes for trying to lyse host cells. One of those was phospholipase C. We looked at capturing human leukocytes using anti-CD45 immunomagnetic separation. So we put together a whole load of methods, but one of the steps that we found was weak was that once we had lysed human cells, we couldn't get high activity of nuclease in whole blood. And so we went looking for nucleases capable of doing that, and we came across the salt-activated nuclease from ArcticZymes, and that was really proving to be quite good. But one of the challenges was to get the buffer right. So we had to try and buffer it so that it would be as active as possible in blood. And so what we did was that we tried out a number of buffers. We tried out the buffer that was provided by the company on the website, and that worked, and it gave us some host depletion, but it was probably only getting us 100, 1,000-fold removal of human DNA. And one day, Solomon came back from the lab, and he was really excited because he'd gotten a kind of a combination of anti-CD45 with some mol lysis digestion of cells and with HL-SAN enzyme to get him 10 to the 6-fold depletion of human DNA. We were all really excited. We went on to combine that enzyme with phospholipase C, drop the other part, and come up with a very nice kind of 45-minute, 1-hour protocol that was able to get us that 10 to the 6-fold depletion. Solomon went on to do some lovely work with that. Then he was towards the end of his PhD. A new postdoc came into the lab, Gemma Kay, and I said, Gemma, you need to get up and running with this metagenomics protocol. It's brilliant. This host depletion step is where we need to start. And she couldn't get it to work. And we did not know why. And she'd made up the buffer, just exactly like Solomon said. And we had to get Solomon back from his write-up, and we had to try and figure out what was going on. And so it turned out, after a lot of investigation and some reverse engineering, we figured out that Solomon, what he'd written on the tube was what a 1x buffer was, but what he'd actually made and what was in the tube was a 10x buffer. So we were adding 5 molar salt and not 0.5 molar salt to the blood, and that had a huge impact on how the enzyme worked and how active it was in blood. So that's how we happened across getting such a highly active and a huge amount of host depletion happening in blood, and that's what we needed for metagenomics to work in an implementable way so that it would not take too long and not cost too much to do sequencing and use that for infectious diseases diagnostics.